maybe, well, that's just because it's stretched out.
Yeah, that's gonna look really good.
Yeah. Well, I'm really bad at ribbing because of the way I was curling, and so I literally just had to do like 2 rows really, really slowly every single night to get the muscle memory of moving my finger from front of yarn to back of yarn.
Okay, floor is open for questions. Anybody have one? We can go to another one. Anybody? Anybody? Just one person?
I sent you an email yesterday about lentiviral vectors.
Sure, why don't you just read the question?
The question that I had about lentiviral vectors, or the question it related to?
Oh, you can do it about lentis.
Oh, okay. So I guess I have multiple questions about lentiviral vectors.
Okay.
The first one, so in class you said that lentiviral vectors are RNA vectors. And I was looking them up more online, and it looked like they can be integrated, but people integrated into a genome, but people also do like another vector, I think, that has like reverse transcriptase.
Yeah, so the version I talked about in class was, let's say, version 1.
Okay.
It involved the removal of the replicase in the coat protein from the virus and an insertion into a suitable mammalian genome. But as the years progressed, people evolved versions 2, 3, and 4, and in particular versions 3 and 4 involve co-transforming the mammalian cells of your choice with 3 different plasmids. One plasmid carries like a surface protein that's a part of the virus, one plasmid carries a replicase, and then one plasmid actually carries the backbone of the virus with the gene of interest inserted into it. So when you transform with 3 different types of plasmids, you can be sure that not every cell gets all 3. But the point they make is that, okay, you're losing some efficiency by only being able to produce viruses in, let's say, 60 to 70% of the cells instead of 100%. At the same time, you choose which mammalian cell line you want to work with. So for some people, that can have a positive impact. So I would say today people are using that approach rather than the stably integrated ones. But they all work the same way.
So my follow-up question to that is, can you only use them in cell culture conditions, or would you be able to use that in multicellular organisms as well?
I got the last, what was the first part?
So can you only use it in like cell culture conditions?
The packaging system? Or the
just
virus?
Lentiviral in general?
No, the virus can work in a multicellular organism.
Okay.
You create them in cell cultures, but once you do, you harvest those viruses. And a lot of them are actually derived from HIV. So we know HIV can certainly infect multicellular critters, and that's why they're using this. So they create these, they're calling them pseudoviruses. They create these pseudoviruses, harvest them, concentrate them, and then inject them into an organism that they wish to study with the introduction of the new gene.
Okay.
Keeping in mind, packaging is an exceedingly specific process. You saw how specific it was with Lambda. It's equally specific with retroviruses, and it's equally specific with these lentivirus type vectors. So although you're having cells, you're creating cells in your Petri dish with 3 plasmids, only your vector gets packaged. The RNA from your vector will get packaged, I should say it that way.
Okay. So you said it would be the RNA, only the RNA gets packaged.
Only the RNA.
So would that mean that when it's introduced into an organism that it would only be transient?
Yes.
Okay.
Yes. I mean, at least for gene therapy purposes, nobody wants to integrate anything if they don't have to.
Okay.
Okay.
2 questions. First one is the gene silencing, the 4 different reasons used to explain why different transgenic multicellular eukaryotes show different levels of transgene expression.
Okay.
Can I run by the 4 reasons that I came up with?
Let's see. Go ahead.
Okay, so the copy number,
Okay.
the location of integration,
Okay.
chimericism, and
Say
then
that last one.
chimericism.
No.
Okay.
We take for granted that the first thing you're going to do is eliminate chimeras.
Okay. So then the next one that I had was methylation.
Right. And that is sort of related in some cases to the place where the gene integrates, but
Yeah.
if we think of RNA-dependent methylation, then it's independent of the place where it integrates.
And then the last one I came up with was the direction of integration. You know, if the gene inserted in the correct orientation or if it was flipped.
Well, the genes we make generally have a promoter at one end and a terminator at the other, so would orientation have an impact?
Probably not.
No. What could?
If it was put in front of a promoter, but
Right.
that would be, again, the location of integration.
Oh, I see. Yeah, I see. For me, okay. Maybe I'm splitting hairs. I would say that integration reflects, in my mind, whether it integrates in silenced DNA, like in telomeres or centromeres.
Mm.
On the other hand, if it integrates near a enhancer or a silencer, then the endogenous enhancer or silencer can influence the expression of the transgene. And if it integrates sort of, let's say, in a gene, but, well, even if it, regardless of the orientation, if it integrates within a gene, you're going to get either what we call promoter occlusion, where the promoter of the gene is here, facing like that, the gene of interest is here, and the polymerases that start here collide with the polymerases that start here and knock each other off. So we call that promoter occlusion. It leads to a decrease in gene expression. And then in conversely, the promoter, the gene of where the target is facing in this direction, the gene of interest is facing in this direction, the polymerases start at each end and they collide in the middle, and and that prevents them from ever making complete transcripts. So I would probably say, you know, any 4 of those descriptions would be fine.
And then my second question was
And you could also say, excuse me, one more thing. It's kind of so obvious that sometimes we ignore it. You can get mutations. Sometimes during integration, you don't integrate the complete sequence. So, but that's, you know, yeah. Okay.
And my second question was also in the gene silencing category was, I just was a little confused what you meant by this question. The presence of methylated Cs in or near a silenced gene does not prove the gene was silenced for that reason.
Okay.
Explain why.
Okay.
I'm not sure what reason is being referred to.
Why doesn't evidence for methylation prove it is the cause of gene silencing?
Okay. So it's saying that just simply because something is methylated, why would it not inherently mean that that's silenced?
Yes, and
Okay.
why wouldn't it? What are the caveats?
Would that be the post-transcriptional silencing?
Oh, that would be transcriptional silencing, but it's silencing. So, but why doesn't the presence of methylation inevitably prove that it is a cause of silencing?
That I forget off the top of my head.
Anything can happen before then?
Sorry?
Anything can happen before then?
Well,
Or
actually, I
not.
can't exclude that, you're right. But can we get anything more specific with regards to methylation versus silencing?
Silencing only occurs if it's methylated at the promoter. So if it's methylated in or near the gene, that's not specific enough.
That's not specific. It sometimes indicates, if you have methylation in the coding sequence, it sometimes indicates that you could have methylation in the promoter region, but it's no proof. And could we even get more specific than that? How do we assay for methylation? Or let me say it differently. How did I say we were assaying for methylation? Because there are other methods than the one I talked about in class.
I think the one that you mentioned was like using restriction enzymes that were sensitive to methylate.
Can you imagine a limitation that that might impose?
You would only be able to work with enzymes that have that limitation. I don't know exactly how many restriction enzymes would be.
There's not so many.
Yeah.
There's really not so many. There are lots of enzymes sensitive to methylation, but the real test requires that you be able to cut the same site, whether it's methylated or not methylated. So you always require a pair of enzymes then. But there are some. But what is the limitation to that method? It's not the availability of the enzymes.
Is it the availability of the sites themselves?
The what?
Of like the sequences themselves?
Yeah. And what about the sequences?
The sequences that the enzymes would be able to recognize.
What about?
Whether or not they were methylated, or
Well, let's say they were, or they weren't, but let's say they were. If you see a site that's methylated, a GATC site or something like that CCGG site, GGCC site, any of those, if you see it's methylated, does that prove that the gene was silenced for that reason? What else has to be true for there to be any kind of serious correlation between methylation and silencing? Between that assay for methylation and silencing. Anybody want to take a stab at it? How do we think transcriptional silencing occurs? I mean, what does this evil methyl group actually do?
I mean, methylation prevents the promoter from recognizing, or not the promoter, the polymerase from transcribing the gene, right?
No.
Darn it.
No, I gotta say no. I would have to discount that answer. Is methylation affecting the RNA polymerase? Or is it affecting something else? So the point is that the RNA polymerase is not so sensitive to methylation per se. What is sensitive are the transcription factors, the enhancer binding proteins. So, I mean, and this is kind of built into the system, you might say. The cell wants to be able to control genes in a number of ways, and one of them involves methylation. What better, in eukaryotes at least, what better than having enhancer binding proteins that can't recognize methylated sequences? Because then you can silence a gene simply by methylating the enhancer. So not the promoter per se, although when we talk in a conversational term, we often say the promoter covers everything, but it really doesn't. That's what we tried to say in this course. No, you need to block the binding of the enhancer binding proteins. If your restriction sites don't overlap the enhancer sites, you're not learning anything other than the fact that in the vicinity of the promoter or in the vicinity of these enhancers, there are methylated sequences. But So that's why the assay that I would describe to you, where you're looking at restriction cutting, has many limitations. It doesn't detect, there's not always suitable sites up there. Those sites don't always overlap enhancers. And so you really cannot make if you see a negative result, you definitely can't say that the methylation is not silencing the gene. And if you see a positive result, you can say it's suggested, but not proven until you know where the enhancers actually are. Okay. It's not really, it sounds like splitting hairs. It's really important, though. So do we have other questions? Yeah.
So also in gene silencing section, the first one, based on our understanding of eukaryotic silencing, why is it better to target siRNA to the 3' end of the gene?
I don't know. Why?
I was thinking it was because the 3' end of the gene tends to be, at least that's where the poly A tail is added onto the mRNA. I don't know.
Okay. Keep in mind, if we are looking at post-transcriptional silencing, the poly A tail is added before we see any sign of silencing. It's all occurring in the nucleus, and we know in post-transcriptional silencing, nuclear RNA production is normal. So all the splicing is going on, all the capping is going on at the 5' end and tailing at the 3' end. The advantage of targeting the 3' end of a messenger with siRNA is that when it binds, it can serve as a primer for RDRP under those conditions in those organisms which have RDRP, which are things like Drosophila and nematodes and plants and filamentous fungi, but maybe not mammals for some reason. So if you target, and probably extension, all vertebrates by extension. So if you target the 3' end and that siRNA becomes a primer, RDRP will bind to the hydroxyl group and then copy the entire length of the mRNA into double-stranded RNA, which will make a huge target for Dicer to come in and cut it up. And so each one of those pieces then becomes a trigger for new RDRP extensions and therefore new dicing. Okay, so you're from that one single siRNA that bound to the messenger's 3' end, you generate 5 or 6 new ones. And then those 5 or 6 new ones can now target genes and again produce a number of new primers. Now some of those will target the 3' end and then we'll get 5 or 6 new primers from that. Some of them will target the middle of the gene and we'll only get 3 primers from that one. And some of them will target the 5' end and we'll only get 1. But in any case, it's a catalytic reaction. We are not losing the numbers of siRNA just because it was used in one reaction. We are using it to generate new ones. Now, if you only targeted the 5' end, you can only go just so far. And so you can never generate more than 1 or 2 new siRNAs from the dicing reaction there. So the more of the RNA that you can make double-stranded, the more new siRNAs will be created to create to target even more copies of that messenger. Okay.
Thank you.
Let me see see if anybody wrote anything. I didn't see anything, but you never know. Somebody could have. Come on. Okay. Anybody here have any more questions? Yeah.
One of the questions, I just wasn't sure if you were talking about something specific or just like in general. The question is, why did cell lines show increased degrees of silencing as the copy number of transgene increased?
Okay.
And I wasn't sure if you were talking about like a specific scenario or...
Well, I was talking especially about this experiment where they were transforming Drosophila tissue culture cells with chloramphenicol transferase gene.
Okay.
And they got a huge number of cell lines. They cataloged them as to how many copies of the CAT gene were integrated. And then they graphed the level of expression versus the number of copies. And what they saw is that in general, the highest levels of expression came from cells with 1 or 2 copies. And the lowest level came from cells with 100 to 500 copies. So that's what I was thinking. Now that principle probably holds true even when we only get 3 or 4 copies integrated. But in the case they had, they had a whole spectrum of cell lines with different copy numbers. And they just saw this nice sort of parabolic decline in level of expression relative to the number of copies were integrated.
Okay. So in that experiment, I guess, I don't think we really talked about if they said why the silencing occurred.
No.
But I was thinking that potentially it would be like post-transcriptional silencing because of the, like, if expression was actually happening. Well, I don't know. I mean, I guess we didn't talk about if things were
We didn't
being
talk about
expressed.
it, but you can extrapolate, so why don't you finish your thoughts?
Well, my thought was if they did experiments with like looking at the RNA levels and they saw that RNA was still being produced, then it would probably be post-transcriptional, and I would assume that it would have something to do or be similar with like RDRP and that process.
Yeah, I mean, and yeah. Maybe you can just flesh out the statement. Why do you think that the number of copies would lead to post-transcriptional silencing?
Well, I think that potentially, like, if they actually showed that they had, which with the graph they did, they showed that they had a lot of copy numbers integrated.
Yep.
Assuming that they actually were expressing, because like, I don't think that we looked at the RNA data, or if they even did RNA.
That expression was, I don't know if that was protein or RNA, but they generally go hand in hand.
Yeah.
So it was either the RNA levels or it was probably the CAT activity, if I recall.
Okay.
So, okay.
So if they were looking at the CAT activity and CAT activity was decreased, I would make the assumption that RNA levels were also decreased, or
Yes.
by some method.
And why would they be decreased? Because.
And my thought is that it would be post-transcriptional.
Yes, and how, and just fill in the gaps.
And I'm thinking that it's post-transcriptional, so Drosophila are invertebrates, and I'm assuming that they have RDRP.
They do.
So I would think that they would be making just tons of the transcript, and that the cell wouldn't like that, or
Ooh,
like.
and how, and what would the cell do when it doesn't like something? I mean, I love, anthropomorphizing things, but in this particular case, the cell's liking or not liking is not necessarily the issue.
Mhm.
What is the issue?
I
Why would excessive amounts of RNA lead to silencing?
I would think that the process, or not the process,
We have models for that from experiments that I talked about later.
Yes, so I would think that the cell would, or
Go ahead.
proteins within the cell would interpret that to mean that there's like an infection going on or something is happening that is making the DNA, or not the DNA, is overproducing the RNA. And
Yeah.
so then there would be processes in place, like
Yeah.
RDRP.
And what
with
processes?
dicers. Like the siRNA and like RDRP.
Yeah,
with
that's really what I
dicers.
was getting to.
Yeah.
In other words, it's nice to be able to make add a specific molecular mechanism
Mhm.
to the story. It is true that the cell downregulates things because it has this capability to do so. But just saying that it downregulated those RNAs doesn't provide us with an explanation. It just provides us with a summary. So what we want to say, maybe, is perhaps the excessive amount of RNA bound back to the template and triggered transcriptional silencing, as the viroid system showed us. Or perhaps it triggered some of it, like the more RNA you make, the more chance you have that some of it will fold back on itself, making a double-stranded RNA that gets diced, and then in turn triggers dicing of all the other RNAs, even those that have not bound back to themselves, formed a hairpin with themselves. So the more RNA you make, so think of it, as an equilibrium almost. You have a probability of an RNA folding back on itself, but it's weak. It's not going to happen inevitably. It's only going to happen sometime. It's only going to last for some time. But the more molecules you produce, the greater the chance that some of those will fold back and remain folded long enough to attract dicer. And once that happens and you start generating siRNA, you don't even care whether they folded on themselves, because those siRNAs will be able to attack the linear messengers. And so you'll just dice all of the cat transcript that way. Okay? So I think, if you have the opportunity to talk mechanistically, I would encourage you to try to do so. You find it's because sometimes, nobody asks for mechanisms. Nobody asks you to learn mechanisms in their courses. Sometimes the classes kind of even say, we just don't know. Let's not worry about mechanisms. Let's just look at the phenomena or what have you. I don't know what they say. But you'll find out that if you can put a mechanism there, you'll find it's possible for you then to come up with hypotheses, how it works, models for how to test it, and then experiments to verify or disprove your hypothesis. And that leads to papers, and well, that's a good thing. Papers are good. Even electronic papers that don't cut down trees. Okay. Any other thoughts, questions? Yeah.
The transposon, the two aspects of gene regulation coming from TN10. That was at the beginning of the semester.
It was, and I wanted you to go all the way back to the beginning.
Yeah. I'm trying to remember.
It wasn't the full beginning.
It wasn't
more-
the full
It was like the first third of the course, or the
Yeah.
beginning of the second third of the course.
Because when we're talking about transposon tagging and transposons, are we talking about like how when they integrate, leaving behind the inverted repeats,
So,
or not?
what did you just say?
They don't leave behind. They leave behind their footprints.
They leave behind footprints, direct repeats.
Yeah, direct repeats.
It's a stretch, I recognize that, but the point is that I chose TN10 in this course because we had learned some different things from studying of TN10. It's not to say we didn't learn things from TN1 or TN5 or TN903 or whatever, but yeah, TN10 gave us some really important things. So the first thing that I talked about was it gave us a convenient regulatory system based on the TET repressor. That even as you saw Monday when I was talking about those cell lines, the studying of cancer, they use that same promoter repressor system, operator promoter system. Now, we knew about operators and promoters in prokaryotes long before TN10 was even discovered. We knew it from the lac operon. But the fact is still that TN10 did give us this particular system, which works much better in eukaryotes than the lac system does. The second thing we got from TN10 was antisense regulation of genes, because you recall that the transposase has two overlapping promoters going in opposite directions. So there's the promoter P-in that starts at the end of the 5' end of the transposon, 5' end of the transposase, and it transcribes the messenger that is then decoded into the transposase. And then there's P-out, which actually lies within the coding region and faces out towards the genome. And people learn that that little RNA made from P-out, of course, because its sequence overlaps the transposase sequence, it would be able to bind to the 5' end of the transposase messenger and block translation. So that was really the first natural example that we had of antisense RNA being used as a regulator. And it turns out there's not too many genes in prokaryotes that work that way, but it did give us a tool that we could use in eukaryotes to easily silence a gene by simply taking the clone gene, cloning it in the reverse orientation in a vector, and then introducing it so that it would be making antisense RNA to the sense RNA that was already present. At the time, we didn't know exactly how antisense works, but I think if you go through the notes, you'll see that it's very likely that antisense works by post-transcriptional gene silencing processes. It is not necessarily an independent process on its own.
Would you also say wings clipped transposons? I guess that's not really gene regulation, that's more
What was the thing that you said?
wings clipped transposons, because we talked about it with transposons, and then we talked about how
Yeah.
you kind of can use similar, at least ideas, when
Right.
working with agrobacterium and with CRISPR-Cas9 of
Yeah.
separating the component parts. But
And how was wings clipped in, I would say that's probably not a suitable example. How was wings clipped in fact discovered? Or not discovered, how was it created? How was it invented?
by separating the different parts that are going to cause your transposon to jump.
And why, what transposon?
Wasn't that TN5?
No.
Darn it.
Why would we call it a wings clipped one?
Oh, wasn't it from one of the Drosophila
Yes.
transposons?
So it was from the P element in Drosophila.
Okay.
And so those guys think they're so funny. So they called that thing, that operation, a wings clipped transposon, because you know what you do to flies, right? So. Okay, but I would say that's not a suitable example of that. That's really a tool for tagging and so on, but it doesn't reveal anything about gene regulation.
Okay.
Or it's not used for gene regulation. No questions? Any questions? Yeah? Chloe?
I guess this one kind of relates to the question I asked previously about the cat gene and like how we didn't know if it was transcriptional or post-transcriptional, or how at least they didn't say if it was transcriptional or post-transcriptional. So another question that you asked was define or describe a definitive experiment that would determine whether it was silenced by transcriptional or post-transcriptional.
What would you think a definitive experiment would be?
So I'm thinking that you would isolate the mRNA from the nucleus,
Yes.
And if mRNA levels are not detectable, then it's due to transcriptional silencing.
Very good. You got
Okay,
it.
so that is.
That's it.
Okay.
That's the basic thing. Don't fall back on methylation, because you've already seen in the best of circumstances how we can't totally rely on methylation. The really big difference actually comes to the words themselves. Transcriptional silencing means no RNA would ever be made. And post-transcriptional silencing means RNA is made, but then it's destroyed. And so if you isolate nuclei and you assay for the RNA there, the heterogeneous nuclear RNA or the primary transcripts, and you see it, then you're not looking at transcriptional silencing. You can't be, because otherwise where'd that RNA come from? And then what happens then is when it gets out into the cytoplasm, Dicer goes after it, as well as some other enzymes. Anybody else want to take some questions?
I do have one more.
Okay.
This one is how long are bacterial transposon footprints? And that one, like, I looked at, like, there's like a table that you had, and it had like different.
Yes,
Okay, so I said between 5 and 10 days per cell.
Yes, that's fine.
Okay. I
Let
wasn't
me-
sure if you wanted an exact number.
No.
Okay.
Let me tell you why. Because when I ask, and this happens every year, but I'm hoping this year it won't happen because I put this question in there, and maybe it'll make people think a little differently. Every year, in the past at least, I've asked the question, describe how you would isolate a transposon tag gene. Every year, somebody, and not just one person, many people, say, well, I would look for the footprints. And if I saw a footprint, it would be an indication that there had been a transposon there. Now, there's a lot of things wrong with that answer, but the biggest thing that's wrong is simply that it's a tiny sequence. And there have been lots of these transposon events over the course of the millennia. And so there's going to be a lot of direct repeats left behind in the genome. And finding one doesn't tell you anything about whether it was a recent event or a past event. And in fact, finding one is really hard because they're just, let's say there are 11, 12 nucleotides. So what if if the cuts are made such that each half duplicates 11 bases, or each half duplicates 6 bases, when the footprint is left, we're going to be looking at a 12 nucleotide or a 22 nucleotide or something like that, 24 nucleotide in direct repeat. Screening for those things would be impossible. I mean, you would literally have to sequence the genome. You wouldn't be able to use probes. You would literally have to sequence a genome in order to find it. So it's totally impractical to say that that's the suitable strategy. If you did want to find a tagged gene, what would you do? You get a Drosophila, you breed it, a P system, using the P system, or even using let's say you use a natural transposon, so it theoretically could jump if it wanted to. No, I don't want to use it. Yeah, okay, we can use a natural transposon. And you pick up a mutant that it has eight legs instead of 6 legs. That's totally impossible. Well, no, it could happen if the mouth parts transformed into legs. So okay, so we have 8 legs instead of 6 legs. And you say, gosh, I really want to get that. So how would you isolate the gene that had been mutated to get that 8-legged phenotype? What would you do? Well, you could simply just say, look, if in today's world, because sequencing is relatively cheap, I'll get a grant, and for a few $1,000s, I'll just sequence the mutant's genome, and then try to compare it with the wild-type genome, and then try to identify a gene that has something like a transposon or a transposon footprint in it. That's possible, but let's say you're not just rolling in money and don't have the time for that. What would you do?
Are you trying to imply using a wings clipped transposon, inserting it into the genome until you get a reversion to wild type, and then finding or relocating the wings clipped transposon using some kind of selectable marker?
No.
No? Okay.
It's a good answer, but no. And the reason is because if a transposon has induced a mutation, it's probably done so by knocking the gene out. If you then go ahead and retransform with a wings clip system, and you happen to get a revertant, it's not going to be in the gene that was initially knocked out. It would be in some gene in that pathway that that compensates for or circumvents the mutation that led to the 8-legged phenotype. So, no, we won't use a new transposon. What could we do? Eric?
Since I'm not too sure about this example, since I believe you're using Drosophila, right? But
It could be TY1.
well, I'm thinking you use like a genomic library of wild type genes, and you slowly introduce one gene at a time. Of course, I don't know if that's easy to do in Drosophila. I'm not familiar. Of course, I know you've explained this during lectures, so I'm just forgetting what the answer is. But I would want to try and introduce the genes one at a time until the wild type is restored in presumably the progeny, because I presumably, obviously, the legs aren't going to go away in the current living one. So like you have to like breed it, or something, and then the progeny will no longer.
People have, so you're using complementation basically, and
Right.
people have done that. It's not necessarily one gene at a time, it's populations of gene, and then they get a positive result, and then they take that population that gave them the real positive result, and then they divide that into new, smaller pools, and they keep dividing the pools smaller and smaller until they're up to one gene. But no. So, but think of it in a simpler way, a much more trivial way. I'm assuming, and nobody said anything, but did you want to try?
I just had one question.
Good.
Do we know that it's in fact because of a transposon, that mutation? And do we know what transposon it is?
Those are the perfect questions to be asking, and I'm going to say yes and yes.
Then
We
can we
know it
use-
was due to a transposon, and we have the sequence.
So presumably that transposon is, well, I guess potentially could be in gene. I guess I was gonna say, you use the transposon as a probe, but I think that's kind of the same issue with using a probe for the direct repeats, maybe?
No, because transposons are big.
Okay.
So if the transposon is around 3 kb.
It's
or maybe-
unlikely that there's a random sequence. that
Oh
matches.
yeah, it's very unlikely. Now there may be multiple copies of the transposon, and that can be problematic, I agree with you there. But yeah, we would make a genomic library. We would take a copy of the transposon that we believe was responsible for the mutation. We make it radioactive. We screen the clones of the library to pull out clones that carry that transposon. And then we sequence the clone, especially looking at the sequence to the left and to the right of the transposon. that sequence to the left and to the right of the transposon should be the gene that's been inactivated, if the transposon jumped into the middle. And so that would be a way of picking up a new gene, as long as you have something defined as a probe. And so, you know, in some cases in this class, we've talked about using the gene itself as a probe to isolate the transposon, for example. And in other cases, we can use the transposon as a probe to isolate the targeted gene. Now, we still have to do experiments, and complementation would be the obvious one to verify that result, that we picked up the right gene. But yeah, go ahead.
One of the issues I had when first saying that answer, or proposing that answer, was I think I was thinking, like, what if we use the transposon jump in the sample that you had, and then so maybe you find it in a different gene.
Yep. That's very true.
And you know, it like already left the gene that it mutated, and therefore that's causing the phenotype you're interested in.
Yep, and we would be really confused. And that's why we needed to make those wings clipped transposons. But in the example that I'm talking about, we're using a natural transposon.
Right.
And you're right. We might not pull up the actual gene that caused the phenotype that got us excited. We may be pulling up a secondary thing that the transposon jumped into afterwards. So you're right. That's why we have to verify that we have the right gene by complementation, by putting a wild type copy, an uninterrupted copy of the gene we've picked up back into the mutant, and see whether it restores the wild type phenotype.
But I guess by doing that, doing the transposon one, using transposon as a probe, you know, you sequence the left to the right. You basically use that to then get the gene right.
Yes, that's right.
And then I guess that at least maybe if it's assuming it's a secondary thing, right? Like it's maybe not the right gene, or it's a candidate, I guess you could say. You would it narrows it down instead of just going from the start, like having a complete genomic library, and then just doing every gene.
Yes,
to like kind of brute
that's
force
right.
in a way.
It's a lot quicker. It would be a
Okay.
lot quicker. Yeah.
So I was gonna say, if we're using a natural transposon and it has jumped, are we then screwed? Is there, aside from sequencing it, is there another way to do it if the transposon has already jumped? and
It's
you
a
can't
funny
find it via
situation
that?
because here we have to reorient ourselves. We are working with a multicellular organism. So when I say that the transposon jumps out, it doesn't necessarily mean it jumps out in every cell. So the chances are high that there'll always be a population behind that actually has the original interrupted gene. But yes, you're right, we would be pulling up multiple genes because there would be multiple places where the transposon had jumped. Yes, we would be screwed unless we can do this complementation to verify the results. Okay. Yeah.
I'm guessing, kind of like a follow-up question to that, since it's just like Drosophila, right, multicellular organism, presumably you can isolate the tissue or the part of the fly that has that phenotype, right? So like,
Yes.
you know, so like you think you briefly mentioned, like if it's the mouth right area that's mutated
Right.
and therefore it turns into legs.
Right.
You can just, instead of using the entire fly as the sample, you know, to get the DNA and to do the probing and stuff,
Right.
you can just grab the parts that are actually mutated, like the area, the region, right?
Right. You could, and
I don't know if that would like potentially help somewhat, like make sure that you increase the likelihood that the presumed genes to the left and right of the probe are actually like the genes responsible for that phenotype.
yeah, you're right. You can do that. It may not be worth the effort because the phenotype itself, this extra pair of legs, would have been determined in the embryogenic state while the imaginal discs were growing. And so once that has been triggered, they could have jumped out because the gene isn't used at a later stage in development. So most of the legs of these mouth parts could actually have the gene in the wrong location, in theory. But I think in most cases, people will simply grind up the whole thing. I mean, I'm trying to think of what they were doing there. There were experiments where people only isolated the heads, but I can't remember what that purpose was. It was for one of the cooler genes, maybe, but I just can't recall what that experiment was. But I think in most cases, people just, dump the things into a blender and,
Okay.
yeah.
So we talked about how the footprints obviously are not big enough for a probe. How many base pairs do you need, sort of minimum, for a probe?
So there are several options for you. Now, if you're doing standard southern hybridization, it's probably advised that your probe be around 200 bases up to 4 or 5 kb, depending on what you're doing. Because that is stable under a variety of conditions. But keep in mind, there are methodologies where people are investigating single nucleotide changes in an organism, especially to identify human disease, inherited diseases, and where one base difference is what's responsible. So in that case, they do have conditions determined which would allow your probe to pair even, the sequences are fairly small, and I can't tell you how small. They're probably they're 20 to 50 bases. They will pair with the mutant allele, but not with the wild type allele. And the only difference is one base. But they're very special conditions, very finicky experiments to do, but they can be done. So you can use very small probes, in fact. I mean, you can use 22 nucleotides in many cases, but you'll wind up trying to, I've used 22 nucleotides, but you'll wind up picking up a whole host of genes. You've got to keep changing the stringency. It's again, not cost effective in terms of the labor. If you can make a big probe, you do. I was using it because I was trying to screen for specific proteins with a specific domain. And that domain wasn't so big, so it was in a cyclin, or it was a CDC type gene. So I wanted to try to pull out that CDC type gene, and but it was just, it really did not work well. So I would say, so you can pull out genes, you can develop conditions. You use 42 degrees, or use sometimes even 37 degrees as the hybridization temperature. Very high salt. Careful with the washes, but then you have to, you just pull up so many candidates because you're working at a threshold where that sequence may be paired perfectly, and then, in some cases, imperfectly, and you're pulling up in all sorts of imperfectly. And so you're pulling up lots of genes, each representing a different degree to which that 22 nucleotide base pair sequence bound. So, wouldn't recommend it. I made enough mistakes in my life, so. What else? Well, if we don't have any questions, we can stop now. People can go home, get some food, some drinks, some edibles, treat your friends. Before you get too wasted, try to fill out the evaluation forms if you haven't done so for all your courses. And I guess I'll see you, is it really next Wednesday? Is it next Wednesday or the week after?
Next Wednesday's finals week.
It's really next Wednesday? Yep. Okay. So I'll see you guys next Wednesday.
Do we need to come here, or were you gonna email us?
Yeah, it's gonna be in this room. Oh, no, I'm sorry.
Okay.
I'm not. Yeah, okay. What we're gonna do is the following. My guess is this. I'm going to send you the test Tuesday, probably like late afternoonish. And then you'll have 26 hours from that point. So if I send it at 4, I would like you to return it by 6 on Wednesday to me. Okay? And these are not meant to be exceedingly long answers. You know, they're not meant to be long essays. You can keep them fairly succinct. And like I said, you'll be able to use any resource that you have available. If you want to work in a group, work in a group. If you want to use your computer, use your computer. It's up to you to decide, you know, what you're going to get out of this exam. But hopefully everybody will get an A. Yeah.
Can we just email you the?
Yes. Email me a Word file, not a PDF. Okay? And I'll have that written in the test. Okay. So far, that's it.
But can we use the same words to define words?
No. That's still verboten.